Hello, good evening to all of you. Myself, Karan Kumar Paswan, and today's I will discuss about order of collections. What are the basically method that are utilized for the collection of sample in the laboratory or hospital sector? So it will be the beneficial to all the students who are pursuing DMLT or BMLT or those students uh, who are taking intern uh, in any hospitals. You can see our video that will be definitely helpful for you. So move to the next slide. So first I would like to tell you the order of sample collections. Sample collections is a process in which we draw the sample from the patients in a particular test is called sample collection. Actually, at first we see the prescription of the doctor, and after that we have to decide the uh, order of sample collections. Okay, at first we have to identify the area of blood collections. This is the area of blood collections as you see on the screen. Okay, I will mark it. This is the area of anticubital fossa. This anticubital fossa, there are various types of vein. Okay, so each vein has a specific region. So you can collect the particular, collect the blood sample from the particular vein. Okay, this is the medial cubital vein, and this is the cephalic vein. This is the bicep bronchi. This is the bicyclic vein, and this is the frontorotates. This is the basilic vein, and this is the medial vein of forearm. Okay, just you can see on the screen, and after that you can easily understand what I am taking telling you. Okay, now uh, now move to the next slide. This is the uh, toolkit, or this is the material, okay, that we are able to that are used for the collection of blood samples. uh, you can see this is the holder, this is the holder through which we collect the blood sample, and this is the needle that are used uh, for the vacutainer, and this is the tourniquet. This is the tourniquet. This is the tourniquet. Okay, and this is the red vial that are used for the collections of by all biochemistry tests like LFT test. You know about that LF LFT test मतलब liver function test, KFT test, LFT test, KFT test, lipid profile, and on all the immunochemistry that test that we have to take the blood sample in red vial. Okay, and this is the blue vial, blue cap vial that are used generally for the blood coagulations. This is the purple vial that are generally used for the collections of all the hematological tests like CBC, platelets, TCDC, hemoglobin, pack cell volume, HCT, hematocrit volume. ठीक है ना? These are the tests which are come under in hematological test. Okay, and this is the heparin vial. है ना? This is the green or heparin vial that are generally used for the heparin test. Okay. And as you see, this is the black vial. This is especially for special test vial that are not used for the general test. Okay, so move to the next slide. Uh, what are the material that are used generally for the collections of blood samples? As you see on the screen, okay, needle, cotton, spirit, disposable syringe. These are the common material, or tourniquet. These are the common material that are generally used for the collections of blood sample. Okay, now uh, now move to the next slide. Okay, order of collections. Why we follow the order of collections? Because it minimize or it prevent from the contaminations. That's why we use the order of collections. Okay, what is the order of collections? Basically, the order of collections is important to ensure accurate test result and to prevent the contamination between the different types of blood tests. And standardized order of draw is recommended by the Clinical and Laboratory Standard Institute. Here is typically order of blood collections. What are the order of collections that we have to utilize on the order? So at first I would like to take the first bottle, that is called blood culture bottle. That is called blood culture bottle. As you see on the screen, blood culture bottle. Why we take the blood culture bottle in the first time? Because it prevents the contamination. Okay, so this is very highly sensitive, and it can be the contaminate because in the blood culture bottle uh, we are able to identify the bacterial infections, or if you contaminate that sample, that means we can't identify the bacterial specimen. So we have to ensure that the we should take the free from contamination sample. That's why we take the blood culture bottle. And now come to the second, light blue top tube. Light blue top tube. That means I am talking about the sodium citrate. That means blue vial. Okay. Generally, in the second time, we take the blue vial container or blue vial tube that contain the sodium citrate anticoagulant. Okay, and this vial are generally used for the coagulation studies. Coagulation studies, that means we study about the prothrombin time, or APTT, activated partial prothrombin time, and all the clotting factor that are used that we have already discussed in previous classes. Clotting factor 1 to 13. Okay, and now come to the third things, that is serum tube. Serum tube, that means red, gold, or tiger top. Red, gold, or tiger top. Okay, these tubes may contain clot activator that are used that are used for the chemistry test. That are used for the chemistry test, serological test, immunological test. When we're talking about the chemistry test, that means in chemistry test we are generally used for the LFT, liver function test, KFT, kidney function test, or lipid profile. When we're talking about the lipid profile, that means total cholesterol, TG, triglyceride, HDL, VLDL. These are the things which are come under in lipid profile. Okay. Now come to the serological test. What are the tests that are come under in serological test? Serological test, that means it is a test which is based on the antigen antibody reactions. What we do identify antibody reactions in the laboratories in serological department? In serology, we have to test like dengue test, IgM. Okay, or uh, dengue test, Vidal test, है ना, and typhoid, है ना. These are the tests which are come under in serological, or you can test also HIV, HCV, HBsAg, है ना, with the help of card test. Immunology test. Some immunological tests that are used for the serum tube. Serum tube, that means either red, gold, or tiger. Okay. Now come to the green top tube. Green top tube, that means talking about the heparin vial. Contain heparin. Heparin is an anticoagulant which is present in the human body. Natural anticoagulant. If question is asked, uh, um, uh, any any examination like government or non-government, name the natural anticoagulant which is so in this condition, so you can write down heparin. Okay, so in heparin, with the help of heparin, you can taste the ABG test. Okay, with the help of heparin, you can test, you can perform the test ABG, arterial blood gas analysis. Okay, now lavender or pink top tube. That means I am talking about the EDTA vial. You all are aware with the EDTA, ethylene dimethyl tetraacetic acid, that contain and used to hematological and blood bank test. In EDTA, when you talking about the EDTA, that means talking about the hematological test. Hematological test, that means TC, DC, HB, है platelets, Paxil volume, hematocrit volume, and RDW, eosinophil, basophil, neutrophil, lymphocyte, monocyte. These are the components which is present in our blood, or if you identify these types of parameters, then we have to collect the blood sample in EDTA vial. Okay, we have to collect in EDTA vial. EDTA, that means ethylene dimethyl tetraacetic acid. Okay, now come to the gray top tube. Gray top tube, that means these are the vial that are generally used only for the glucose test. Only for the which type of test? Glucose test. Glucose test, that means you can test random blood glucose, random blood glucose fasting, blood glucose PP postprandial, or glucose tolerance test GTT. These type of tests, if come in your lab, then you can collect the blood sample in gray top tube, gray top vial. Okay, then you are able to identify the glucose test. Now come to the these are the figure which show the order of collections. Okay, at first we take the blood sample in the uh, blood culture vial. These vial are looking like this type of things that you are able to see the on the screen, and after that we have to take the sample in Blue vial, that means coagulation test, and then red vial for biochemistry test, and purple vial for special test like LH, FSH, luteinizing hormone, follicle stimulating hormone, है ना? And PCT, procalcitonin. These are the special tests that are used generally for the gel vial, okay, or SST, serum separating tube vial, or special tube, okay. And now come to the pur purple vial. Purple vial are generally used for the hematological test, and these are the heparin vial. And now come to the uh, gray vial. Gray vial are generally used for the blood glucose test, or in blood glucose test we test the blood sugar random, blood sugar fasting, blood sugar PP, blood sugar postprandial. These are the tests that are used gray vial. So this is the whole session that we are talking about the order of collections. Okay, this is very easy to understand. Okay, and have you any problem, you can contact in the email ID, or you can WhatsApp. Okay, thank you. Bye bye.